Good morning. Let's discuss on glycolysis. Glucose is the preferred source of energy for most of the body tissues, and the brain cell derive the energy mainly from the glucose. And when the glucose metabolism is deranged, the life-threatening condition may occur. A minimum amount of glucose is always required for the normal functioning of the body. And glycolysis is a pathway which is it is being evolved in nearly all types of organism, and another name for this process is Embden-Meyerhof pathway. So glycolysis is definition wise, the glucose is split into three carbon molecule pyruvate molecule under the aerobic conditions or the lactate under the anaerobic condition, along with the production of a small quantity of energy. And all the reaction steps takes place in the cytoplasm of the cell. Before we start with the actual glycolysis pathway, let us summarize a few important points regarding the significance of glycolysis, as it is the only pathway that is taking place in all the cells of the body, in nearly all the tissues, and glycolysis is the only source of energy in the erythrocyte or RBCs. And during heavy exercise or strenuous exercise, when the muscle tissue lacks the enough oxygen, the anaerobic glycolysis form the major source of energy for the muscles. And the glycolysis pathway may be considered as a preliminary step before complete oxidation of glucose. And this pathway also provides carbon skeleton for the synthesis of non-essential amino acid, as well as it generates the glycerol, which is important component of fats. And as far as the entry of glucose in the body is concerned, in the cells is concerned, so it is being mediated by GLUT4 transporter channels, which are available on all the skeletal muscles and peripheral tissues. And these GLUT4 channels are under the control of insulin. So after having a meal, when the concentration of glucose in the blood raises, so under the influence of insulin, this GLUT channel gets open and facilitates the entry of glucose among the various tissues. So once within the cell, the cytoplasm, this glucose is undergoing the around 9 to 10 steps and converted finally into the pyruvate under the aerobic condition, while under the anaerobic condition it converts up finally into the lactate. So let us see one by one. Glucose under the influence of enzyme hexokinase, and it converts up into the glucose 6 phosphatase, and one glucose 6 phosphate, and one ATP is consumed here. And this hexokinase required the molecular presence of magnesium 2 plus ion. And then next step, the glucose 6 step converts up into the fructose 6 phosphate under the presence of enzyme isomerase, that is the 5-phosphohexose isomerase. Then in step 3, the phosphofructokinase facilitates the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. And in this step also 1 ATP is consumed, and this also requires the presence of magnesium 2 plus ion in the step 3. So in step 3, these three steps are preparatory step. Then next step is next come the splitting phase, in which the fructose 1,6 bisphosphate is broken up into two important component, three carbon component. One is the glyceraldehyde 3 phosphate, another one is the dihydroxyacetone phosphate or DHAP in brief. And this reaction is influenced by the enzyme aldolase. And glyceraldehyde is an isomer of dihydroxyacetone phosphate. So under the influence of enzyme isomerase, it can be converted up into DHAP and vice versa. Then further, maximum duration of time, glyceraldehyde is going to be converted finally into the pyruvate. So this glyceraldehyde 3 phosphate is converted up into 1,3 bis hydroxy bis phosphoglycerate. 1,3-bis phosphoglycerate under the influence of enzyme glyceraldehyde 3-phosphate dehydrogenase. So since dehydrogenase is here, so reducing equivalent NAD is consumed and it's converted up into the energy equivalent, that is NAD2H. And then this in the next step, the 1,3-bis phosphoglycerate is converted up into the 3-phosphoglycerate under the next category of enzyme, that is the kinase, and that is 1,3-bis phosphoglycerate kinase. And here in this step, one ATP is being produced right from ADP. So energy is also generated here in the form of ATP. And in the previous step, the NADH is being generated, which are in the electron transport chain can be converted up into 2.5 equivalent of ATP. So both these are beneficial steps and they are energy generating steps. Then in the next step, the 1,3-bis phosphoglycerate is converted up into the 3-phosphoglycerate, and after the 3-phosphoglycerate, it converts up into 2-phosphoglycerate. It is only shifting of phosphate group right from third position to second position of the glycerate. So it is just an isomerization, and so the enzyme is phosphoglyceromutase. Mutase is also a type of isomerase. And then in the next step, the 2-phosphoglycerate is converted up into the phosphoenol pyruvate, and which requires the enzyme enolase and the magnesium 2 plus ion. Further, the phosphoenol pyruvate is converted up finally into the pyruvate if the conditions are aerobic. So this is how in the cytoplasm the pyruvate is generated under the influence of enzyme pyruvate kinase, and magnesium 2 plus ions are required here. Here also there is a benefit of 1 ATP. Okay, so this still here, these are the steps during the aerobic glycolysis. Once the oxygen is lacking and there is no availability of oxygen or very less oxygen is there, then pyruvate, instead of converting to acetyl coenzyme A for the to join the Krebs cycle, it better is converted up into the lactate. So because oxygen is not there, so pyruvate cannot be further processed in absence of oxygen in the Krebs cycle, so it will be diverted to the synthesis of lactate in the cytoplasm. The enzyme is lactate dehydrogenase, and NADH. Here the NADH which was being produced, it is being required here, consumed here, and that is converted to NAD. This NAD can further be utilized here in this step 5, and it can be reutilized for generation of NADH. So there is total no benefit of NADH here in this step. So this is about the aerobic and anaerobic glycolysis. Now, energy yield, the number of ATP generated per molecule of glucose in glycolytic pathway under the anaerobic condition, that is C first. So these are the various steps. Step 1, which is being channelized by hexokinase. Here there is a consumption of 1 ATP. Then the second step 3, where the phosphofructokinase is working. So there is also consumption of 1 ATP. So there are 2 ATP are being consumed in this step. In the step 6, the 1,3-bis phosphoglycerate kinase is required, and this in this step 2 ATPs are being generated. So this is a beneficial process. And then in the energy yield per molecule of glucose in the glycolytic pathway under the aerobic conditions. So here you have seen here the hexokinase and phosphofructokinase steps. Here also they require 2 ATP. So there is a loss or consumption of 2 ATPs. Now let us see where there is a generation of ATP. So glyceraldehyde 3-phosphate dehydrogenase enzyme, it generates 1 NADH, which is equivalent to 2.5 into 2 ATP, because glyceraldehyde and DHAP. So DHAP also convert up into glyceraldehyde. So like you have seen in the cycle, which is when one glyceraldehyde is consumed and giving rise to one NADH, but it is actually the two molecule of glyceraldehyde 3-phosphate. These are the three carbon species. So total benefit of from one glucose in this step, total benefit of 2 NADH. So that is equivalent to 5 ATPs. Then in the step 6, 1,3-bis phosphoglycerate kinase, it produces 2 ATPs because there are two conversions right from glucose. And in this step, the 1,3-bis phosphoglycerate kinase is converting this glyceraldehyde 3-phosphate into 1,3-bis phosphoglycerate kinase. Then in step 9, which is being channelized by pyruvate kinase, where there is a conversion of phosphoenol pyruvate to pyruvate. So it also produces one ATP, or we can say for 2-glyceraldehyde 3-phosphate, there is production of 2 ATPs. So total from one glucose, there is 5 to 7 to 9, and total 9 are gained and 2 are these two ATPs are utilized. So total gain of ATP in the aerobic condition is 7 ATPs, while in the anaerobic condition, only the 2 ATPs are generated. So this is the bioenergetics of glycolysis process, both in aerobic and the anaerobic condition. Now in this slide, we'll see the what is the difference between the hexokinase and glucokinase. Actually, hexokinase is the enzyme which is required in the first step of glycolysis, which converts glucose into glucose 6-phosphate. And this is the glucokinase. It is one of the isoform of hexokinase. So what are the basic differences? Glucokinase can only convert glucose into the glucose 6-phosphate, while the hexokinase is the generalized enzyme which can convert other hexoses as well into their corresponding phosphates, like fructose can be converted to fructose 6-phosphate, or galactose can also be converted up into galactose 6-phosphate. So what is the line of difference? Occurrence, as far as occurrence is concerned, hexokinase is present abundantly in all the tissues, while the glucokinase is only in the liver, while the Km value is 10 to the power minus 2 millimoles per liter, while the glucokinase, the Km value is 20 millimoles per liter, and affinity to substrate for hexokinase is very, very high, and for glucokinase it is very low. Then specificity, it acts on glucose, fructose, and mannose, while the glucokinase is very specific only to glucose. Induction, it's not induced, but this glucokinase is an induced by insulin and glucose, presence of glucose. Then functions, even when the blood glucose level is low, the glucose is utilized by the body cell. And while glucokinase act only when the blood glucose level is more than 100 milligrams per day. So then glucose is taken up by the liver cells for glycogen synthesis. Move on to the regulation of glycolytic pathway. So as far as hormonal control is concerned, so two major antagonistic hormones are all time working in order to regulate the concentration of glucose in the blood. So it is the insulin which favors the glycolysis, while at the same time the glucagon inhibits the key glycolytic enzyme. Further, the insulin activates the key glycolytic enzymes like glucokinase, phosphofructokinase, pyruvate kinase, etc. While the glucocorticoids also inhibit the glycolysis, and it favors the gluconeogenesis. Then the step 3, that is the phosphofructokinase, is the main rate limiting enzyme for the glycolysis, as it is the slowest step. So all kinetics of this pathway basically dependent onto the working of phosphofructokinase and how it is being regulated. So major regulator of this phosphofructokinase is are ATP, citrate, AMP, and fructose 6-phosphate and all. So let's see how The phosphofructokinase is allosterically regulated by the enzyme. And then ATP is allosteric inhibitor. ATP is the allosteric inhibitor of this enzyme, while citrate is also another inhibitor. AMP acts as an allosteric activator, means the presence of ATP, AMP in the system indicates the lack of energy. It induces or activates the phosphofructokinase. Then fructose 6 phosphate also increases the activity of this enzyme, because if more is the concentration of phosphate for fructose 6 phosphate, it will further facilitate its conversion to 5 fructose 1,6 bisphosphate. So that's how it increases the activity of phosphofructokinase. So regulation summary: three reversible steps catalyzed by hexokinase. That is the step one. Then step three is for being catalyzed by phosphofructokinase, and step nine, which is being catalyzed by pyruvate kinase. So these are the three irreversible steps, and these are the main regulatory enzyme. Out of this, the rate limiting enzyme, as I've already explained, that is the phosphofructokinase. And besides, the two important hormones are insulin and glucagon. Beside other hormones like glucocorticoids, adenocorticoids, thyroid hormones, they too have some influence onto the inhibition of glycolysis, pathway. Now, enzymes, what are the enzymes, how they are being activated, how they are inhibited? So, hexokinase is being inhibited by glucose 6 phosphate. If there is an excessive quantity of glucose 6 phosphate, so through it is the first step conversion of glucose to glucose 6 phosphate, and if it is in excess, so it will through feedback inhibition, it will inhibit the activity of hexokinase. And then glucokinase is activated by insulin while inhibited by glucagon. Then phosphofructokinase, as I've already told you, insulin is activating, AMP is activating, fructose 6 phosphate is activating, and fructose 2,6 bisphosphate indirectly activating the phosphofructokinase. And PFK, phosphofructokinase 2, is also, all these are activator of phosphofructokinase. Through all activation is allosterically done. Then what are the inhibitors? The glucagon is inhibitor, ATP, citrate, and cyclic AMP are the inhibitors, allosteric inhibitor of phosphofructokinase. Then pyruvate kinase, which is a step 9 enzyme, which is meant to convert the phosphoenol pyruvate to pyruvate. It is being activated in the presence of insulin, fructose 1,6 bisphosphate, while the presence of glucagon, ATP, and cyclic AMP inhibits the activity of this enzyme. Now move on to the clinical aspects. Lactic acidosis is one of the important manifestation of excessive anaerobic glycolysis, and which happens due to the deficiency of oxygen. So normal value of lactic acid in the blood is 4 to 15, and once it goes beyond the normal range, it starts accumulating into this lactic acid starts accumulating in the blood and muscles tissues, causes lot of side effects. As in the mild forms of lactic acidosis have been observed in during the strenuous exercise, shock, hypoxia, respiratory diseases, and cancers. In the severe form of lactic acidosis, there is impairment and collapse of circulatory system, because there is the total no impairment of the maintenance of pH. Then myocardial infarction is, then pulmonary embolism, uncontrolled hemorrhage, severe shocks. So alcohol abuse, diabetes mellitus, and mitochondrial cytopathies. In all these conditions, the drastically raised level of lactic acid has been observed. And second condition is the cancer, because cancer cells they are solely dependent for their growth and develop onto the glucose via exploiting the glucose by the glycolysis process. So the cancer cell they lead to increased uptake of glucose via glycolysis. Then blood vessels unable to supply adequate oxygen. So during the hypoxic condition, the cancer cell prefer the anaerobic glycolysis, and that is also known as hypoxic glycolysis, because there is a vast number of small cells, uncontrolled growth of the cancer cell, and all of them grow very fast, and for their growth and development they require no a huge amount of glucose. So involvement of hypoxic inducible transcription factor is there, which controls or the glycolysis, anaerobic glycolysis in the cancer cell. And then treatment lies onto the use of drugs that inhibit the vascularization of tumors. And another way is to cut off the intake of carbohydrates, so that will also control the growth of cancer cell, because they are solely dependent on the glucose through glycolysis pathway for their energy needs. Then deficiency of glycolysis and enzymes. These conditions are rare, but out of which the pyruvate kinase deficiency and hexokinase deficiency are comparatively common, and they lead to hemolytic anemia because the energy depleted and RBCs are destroyed. Then next condition is pyruvate dehydrogenase and thiamine deficiency. Pyruvate dehydrogenase is the enzyme which is responsible for conversion of pyruvate into acetyl coenzyme A in the mitochondria. And this enzyme is dependent on thiamine or vitamin B1. So PDH requires thiamine pyrophosphate, that is the active form of vitamin B1. This explains the serious afflictions in beriberi due to the thiamine deficiency. And TPP deficiency in alcoholism causes the pyruvate accumulation in the tissue, and that results in accumulation of lot of lactic acid and leading to lactic acidosis, because pyruvate is now diverted to the synthesis of lactate instead of acetyl coenzyme A in the mitochondria. And so inherited PDH deficiency may also lead to lactic acidosis. Then Warburg effect, what is this effect? Because in this, there is an enhanced conversion of glucose to lactate in the cancer cell, even in the presence of normal level of oxygen. So this effect has been observed, and that's been by Warburg, and that's how this is known as Warburg effect. So we see the various other effects like Pasteur effect. Pasteur effect is it is the actually the inhibition of glycolysis by oxygen. In the presence of oxygen, the glycolysis has been found to be inhibited. So under the aerobic condition, and that effect has been observed by Pasteur. That's how it's named as Pasteur effect. And this inhibiting effect of oxygen onto the fermentation process by facultative anaerobic bacteria has also been observed, and it is the sudden change from anaerobic to anaerobic process. Then inhibition of glycolysis, like in the under the during the aerobic conditions, the TCA cycle get activated. So there is a lot of synthesis of citrate, and excessive amount of citrate leak back into the cytoplasm that inhibits the phosphofructokinase enzyme, and that's how the glycolysis is discouraged, and there is a decreased rate of glycolysis. The next effect is the Crabtree effect. It is the phenomena of inhibition of oxygen consumption by the addition of glucose to tissues having the high aerobic glycolysis going on. So here you see here, there is an inhibition of oxygen consumption in the presence of glucose, right? So this is the Crabtree effect, and the decreased cellular respiration is caused by the high concentration of glucose. More glucose, more energy, and increased concentration of ATP and ADP ratio as ATP is increased, so low consumption of oxygen. So initially more oxygen, initially more glucose. So that is going to increase the more glucose oxidation, thereby the increased consumption of oxygen. So that's how this is the region for presence of glucose and inhibition of oxygen consumption by addition. That is the Crabtree effect. Now, Rappaport leubering cycle and the significance of 2,3-BPG. Actually, the Rappaport leubering cycle is also known as the shunt pathway of the glycolysis or the supplementary pathway. So it has been observed in the erythrocyte, and actually this leads to the synthesis of 2,3-bisphosphoglycerate and oxygen transport. So we'll see it here. This is the in the normal glycolysis, the conversion of 1,3-bisphosphoglycerate happens via the phosphoglycerate kinase into the 3-phosphoglycerate under the normal condition, and there is a production of 1 ATP. While it has been observed in basically in erythrocytes, there is enzyme 2,3-bisphosphoglycerate mutase. There is a diversion of this normal pathway to like via production of 2,3-bisphosphoglycerate, means the 1,3-bisphosphoglycerate is converting up into the 2,3-bisphosphoglycerate via this enzyme 2,3-bisphosphoglycerate mutase. Further, this 2,3-bisphosphoglycerate via the enzyme 2,3-bisphosphoglycerate phosphatase converts into the 3-phosphoglycerate. So it's kind of shunting between these two substrates via the 2,3-bisphosphoglycerate. That's how this pathway has been named as 2,3-BPG pathway, or it's being for the first time being reported by Rappaport and leubering. That's how it is also known as Rappaport leubering cycle. And then further steps goes in a normal man in the glycolysis. And what is the significance of this pathway, and what is the role of the production of this 2,3-BPG? Actually, the synthesis of 2,3-BPG is like without it happens without the synthesis of ATP. So in this step there is no production of ATP, while in the normal path there is a production of 1 ATP. So this process, one thing, it helps to dissipate the energy which is if it is not needed by RBCs. Then it supply more oxygen to the tissues during the low oxygen condition or in the hypoxic condition. So in the hypoxic condition, this pathway is preferred. So during anemia, high altitude, and even in the fetal blood hemoglobin circulation, COPD diseases, the concentration of 2,3-BPG has been found to be raised. So it has a lot of significance in the biological system. 2,3-BPG actually it has a lot of affinity to the normal. It has a lot of 2,3-BPG combines with hemoglobin, and it reduces the affinity towards the oxygen. So in the presence of this 2,3-BPG, oxyhemoglobin will unload oxygen more easily in the tissues, right? Then under the hypoxic condition, the 2,3-BPG concentration in the RBC increases, thus favoring the release of oxygen to the tissues, and even when the concentration and the pressure, oxygen pressure is very low. And third is the compensatory increase in the 2,3-BPG in high altitude. It favors the oxygen dissociation, and BPG is increased in fetal circulation also. So in this shunt pathway, there is no ATP is being generated. So these are the following point of significance or advantages of 2,3-BPG.